he serves as the associate editor for chemometrics in Willis Encyclopedia of Analytical Chemistry. He is the past coordinator of the chemometric group of the Italian Chemical Society and the coordinator of the chemometric study group of UChems. And he is organizing a very important event next year in 2025, and that will be the 22nd International Conference on Near Infrared Spectroscopy, NIR 2025, and he is the general chair of that conference, which will be held in Rome during July 8 to 12. So with this brief introduction, I would now request to Professor Federico Marini to deliberate on his topic. He has chosen the topic as Analysis of Spectroscopic Data from Designed Experiments with ASCA and Related Methods. So thank you, Professor Marini, for joining here. I request you to please start your webinar.
So first of all, I would like to thank you, Dr. Pandya for the invitation, of course, both to the conference as a speaker and to this very good series of seminars. I was quite happy to see like the idea of having this road to the conference, to this internet webinar, and I'm happy to contribute. And in order not to, of course, to repeat things that would be like the topics of the ANS talk, I was thinking that it could have been nice to like do sort of like a tutorial lecture on some techniques that were developed originally in the field of metabolomics and that I do particularly like, that are the techniques for the analysis of multivariate data that come from design experiment. I was realizing that given, let's say, the time constraint of the lecture, I will mostly focus on ASCA. That is the most used actually of these techniques. But of course, there are also some other similar methods that have been developed in the recent literature. Okay, here. So the first thing that is worth being considering that it was actually one of the driving forces of the development of tools for the analysis of design data is that actually when already at the time of Fisher, so around 100 years ago, when the concept of rational design of experiment was developed, was already evident that it's not enough to properly design the experiment, because of course, if you properly design the experiment, but then if you don't have tools like data processing tools that are, let's say, compliant to the design of experiments that have been developed, then probably one may underuse the advantages of the experimental design. And one can come to as inconclusive results as one could have by not using the experimental design and then go further with the traditional data analytical method that could be available. So in this context, actually, he uses this, let's say, this sentence that statistical procedure and experimental design are like two different aspects of the same whole, like two sides of the same medal in a way. And this is the whole that leads to the improvement of knowledge by experimentation. So why properly designing experiments? Normally, in the context of chemometrics, but not only, an experimental design is used with a twofold aim. This twofold aim actually is the same aim that can be developed up to a different level or up to a different maturity stage, depending on the possibilities of conducting experiments, on the time and cost and resources that one can put into these experiments. So the first level is just to have a qualitative understanding of the process or of the methods or of the system. And the second stage is to get also a semi-quantitative or a quantitative estimate of what is going on. So at a qualitative level, the idea is that I suspect that one, but possibly more factors that can be either qualitative or quantitative in nature, like adding some catalyst or fixing the temperature at a certain level, or having some of the additives at a certain concentration may have on one or more experimentally determined responses. So in this case, the first level is just, I can say based on the experiments that I'm conducting, yes, this particular factor, this particular term has a significant effect on my response, or no, actually, when I am changing the experimental condition, I get a variability that is comparable to the variability due to random error, so to the variability due to the repeatability or reproducibility conditions of my measurement. If I may invest time, money, experiment, resources to get a higher number of experiments, probably I can even have enough degrees of freedom in case I deem some of these effects to be significant, to try to model this effect in a quantitative way. So to try to model the relationship between the responses that I am monitoring and the factors that I am controlling. So far, these are the aims, but where the rational pianification, the rational design of experiment enters, it enters at the level that, of course, I want to do these two, I want to satisfy these two objectives, performing the minimum number of experiments, but at the same time performing these experiments in some specific experimental condition that will guarantee that the estimate of my qualitative or quantitative model are as performant, as good as possible. So you see, it's an ambitious aim, the one that we want to carry out. And for doing this ambitious aim, we need to properly map the experimental domain. The experimental domain is the range of possible values of the factors that we want to investigate. So first of all, we have to be clear about which factors we want to investigate, which experimental condition was potential effect on the response we want to investigate. Then, what is the range of variability at which we want to investigate this experimental condition? And eventually, of course, based on that, and based on the idea that we would like to have an as rich response as possible. So we want to be able to estimate, for instance, the significance of the effects as properly as possible, or we want to estimate the response surface, so the relation between the y and the x with the minimum uncertainty, and with the minimum uncertainty not only on the model parameters, but also on the model prediction, then we will decide where to carry out the experiments. For this talk, we will focus specifically on the first step of this analysis. So of the possibilities of understanding whether one or more factors potentially have an effect on a multivariate response, that in our case would be our NIR spectra. And in order to do so, we are supposing that data are collected according to one specific experimental design. This is not the only experimental design that is possible, but is the kind of experimental design according to which all the theory about the analysis, the analysis of variance and ASCA, that is one of these possible multivariate generalization, was originally developed. I said it was originally developed because, of course, that can be extended to more complicated design, different designs than the one that we will be seeing. But for the moment, we will be focused on the kind of designs that are called full factorial design. Full factorial design means that I decide to control a certain number of factors. So factor 1 here could be temperature at which I conduct the experiment. Factor 2 could be time. And factor 3 could be the concentration of one particular additive that I can be interested in controlling. And if I am assuming that all the experiments that I will conduct will involve only two possible values of factor 1, that means two possible temperature values that I am investigating, 4 possible values of time and 3 possible values for the concentration of the additive. These possible values that the control factor can take are also deemed levels for that particular factor. Then in a full factorial experiment, I am considering that all the possible experiments that I'm carrying out stem or arise from the combination of the investigated level of the factor. So I can carry out of an experiment like the one, I don't know if you can see it, I put the, see. No, it's not visible, sorry. Okay, but so this experiment of the lower left corner of the cube here represents the experimental domain. That is the combination of the range of factor 1, the range of factor 2, range of factor 1, the range of factor 2, and the range of factor 3 here. Then I will end up in conducting 2 times 4 times 3. That means 24 experiments. And these 24 experiments are the combination of all the possible combination of the levels of the investigated factors. If I can take all the possible combination of the levels of the investigated factor, this is called the full factorial design. And for the full factorial design, if I want to understand whether factor 1 or factor 2 or factor 3 or whatever, and their possible binary or ternary interaction, that means the possibility that the effect of one factor on the measured response depends on the level at which the other factors are. That means if I change the temperature at time 0, I will have a different effect with respect to if I change the temperature at time 30 minutes. Then if I have a single measured response, for instance, like the pseudo absorbance recorded at a fixed specified wavelength, then the variability that I observe across the experiments can be partitioned according to what is called the analysis of variance. The analysis of variance is a way of apportioning the contribution to the total variability to the factors and the interactions that result from the experimental design. So here, if I consider only 2 factors, A and B, and a single response X, then the individual measurements that I have, Xij, that means the individual measurements that I collect when I am checking the kth replicate experiments that I do when factor A is at level I and when factor B is at level J. For instance, when temperature is at level 20 degrees and time is at level 30 minutes. Then the signal that I observe can be partitioned according to a general offset. This is what here is summarized by the letter mu. A contribution that accounts for the variation of the signal depending on the fact that I am changing the value of factor 1. So this alpha I. An additional contribution with respect to the general offset depending on the fact that my second factor is at the level J. A possible further contribution with respect to this that we call the main effect of the factor related to the fact that there is a possible interaction between the 2 factors that I'm controlling, this alpha beta ij. And then this would be my model. This would be the idea that I can reasonably apportion the variability due to the control factor in this additive series of additive contribution. Of course, there is something that remains outside this general linear additive model. That is this epsilon ijk that are the residual, is the variability that I cannot account for based on the design. So there is a part, my idea is that there is a part of the variability that I observe changing from one set of experiments to the other, that I want to, let's say, apportion to the experimental, that I want to ascribe to the experimental design. And there's another part of the variability that is the natural variability due to the random factor, random error contribution that makes so that if I take repeated measurements, of course, I'm not expecting to have the same result. Then my question is, the variability induced by the different factors and by the interaction, is it significantly different than the one that I can ascribe to the random error or not? And if so, then I can interpret the sign or the direction of this difference. Here, of course, you see that if I want to be able to separate the contribution of a potential higher order interaction, here the alpha beta, from the random error, at each combination of factor, for instance, 20 degrees of temperature and like 30 minutes of time, I have to conduct at least replicate 2 experiments, because otherwise I will not be able to disentangle the higher order interaction contribution from the random error term. But with these premises, the idea is that, okay, I have an overdetermined set of conditions, so I need to fix some constraint. Otherwise, I'm not able to infer from the experimental data and from my knowledge of the experimental design the value of this additive model. So the mu, the alphas, the betas, and the alpha beta terms. Normally, we need constraints. These constraints is that I will consider the mean of the signals collected across the experiment as my reference, so as the mu here, as the offset with respect to which I am consider the relative variation induced by the factor. This means that the alpha are centered, the beta are centered, and the alpha beta terms are marginally centered. And then I can estimate this term from the data as a series of calculation of means calculated in different ways. Why I am doing this calculation? Because my final aim is to understand whether my changing my temperature produces a variation in the signal, in this case at the absorbance at a fixed single wavelength, that is higher than the variability that I can ascribe to the random error. To do this, normally, I make a significant test for each of these contributions, the one of factor 1, the one of factor 2, and the one of the interaction, that is done by calculating an F-test. Okay, this was the premises, standard univariate statistics, Fisher, ANOVA, around 1920s, 1930s. Okay, but then what if we, instead of collecting a single signal at each cell unit of the experimental design, across our design experiments, we collect a multivariate signal, for instance, a spectrum. Then, instead of having a single value of X for each experimental condition, we will have a vector X, for instance, a whole spectrum collected at each experiment. And each experiment would be characterized by a certain value of factor A, a certain value of factor B, and so on, depending on how many factors I am considering. So, what is the basis of any generalization of analysis of variance to the multivariate case? Let's consider a simple toy example, like the one that is depicted in these slides, where we are dealing with 2 factors, each factor controlled at 2 levels. So we have potentially 4 experiments in the experimental design with the symbols plus and minus here. I am just simply referring to the highest value of highest level of factor A and lowest level of factor A, or highest level of factor B and lowest level of factor B. For instance, if I am considering temperature as a factor with 20 and 30 degrees at level, plus would mean 30 degrees, minus would mean 20 degrees. If I'm considering factor B as time with 0 and 30 minutes as level, minus would mean 0 and plus would be 30 minutes. So this is just a quick symbol here in the scheme. Then, of course, if I have 2 factors at 2 levels, possible combination in a full factorial design are 4 experiments. That would be minus minus, minus plus, plus minus, and plus plus. Here I am assuming, in order to be able also to estimate the contribution of the interaction, to have 8 experiments, meaning that I am repeating the whole experimental design twice, or that each cell, each combination of levels of the experimental design, I am conducting a replicated experiment. So I do my 8 experiments, and I am collecting the spectra corresponding to these 8 experiments in a matrix. Then, how do I proceed in extending the ANOVA, the analysis of variance, to the multivariate case? The first step is to apportion the total variability to the contribution of the different factors and interactions that are accounted by the design. And then what is left out of this apportionment would just be the residual, so the variability that I am reasonably assuming to be ascribable to the random error or to the non-modeled contribution. So the variability not related to the experimental design. And how do I do the apportionment? Exactly as I was doing in the univariate case. Indeed, the first step of the multivariate ANOVA decomposition is just to do a univariate ANOVA decomposition for each of the single columns, so for each of the single variables that make the multivariate signal. So, but then this I can see it already with the multivariate eye. So I take the data matrix, and first of all, in order to account for this offset, that for each variable would be a specific mu, that is the mean of all the experiments for that particular value, then I do mean centering. So my XC is my data matrix that collects the spectra at these 8 experiments, that is first already subjected to mean centering. So the centered data then are decomposed according to the analysis of variance. So how do I do the ANOVA decomposition in a multivariate way? Here, for instance, I want to account for the effect of factor A. The effect of factor A, it means that with respect to the mean signal, I have a contribution, an additive contribution, depending on whether my factor A, we said temperature, is at 30 degrees or at 20 degrees. So this means that in half of the experiment, the first 4, where the temperature is at 30 degrees, I will have the same contribution, and this contribution would have to account to the fact that my temperature is 30 degrees. So I will have a sort of additive spectra that would account for the fact that my temperature is higher. Similarly, in the second 4 experiments, I will have an additive contribution accounting for the fact that my temperature is at its lowest value. And similarly, it would be from the effect of factor B and from the binary interaction AB. The thing is that, okay, but how do I calculate this contribution? Well, this contribution actually is given if we do all the ANOVA theory, by calculating the mean spectrum of these 4 experiments, the red ones, where my temperature was at its highest level. And this mean spectrum will be copied identically in the first 4 rows of the matrix. Similarly, I take the last 4 rows of the centered matrix XC, I calculate the average of these 4 spectra, and I substitute this average spectra in each of the 4 last rows of the matrix XA. So the matrix XA will be made of 4 replicates of the mean spectrum calculated in the first 4 experiments, and 4 replicates of the mean spectrum calculated in the second 4 experiments. And analogously, it will be done for the other matrices. Here you see it with an example having only 3 variables. So 3 variables, and I make a design with 2 factors. Factor 1 is at 2 levels. Factor 2 is at 3 levels, and I have 2 replicates for each design unit. So if you want to see it graphically here, we will have factor A will be 1, 1, 1, 1, 1, 1, 2, 2, 2, sorry, 2, 2, 2, and factor B would be 1, 1, 2, 2, 3, 3, 1, 1, 2, 2, 3, 3. So 12 experiments, 12 spectra in this matrix. These are the raw data. I put a very simple example, then we will complicate it by looking at real spectroscopic data. So from this data, I calculate the overall mean to do mean centering. So, and I subtract this mean spectrum or mean signal for each of these 12 profiles. And I obtain a mean centered data. From this mean centered data, I calculate the mean of the first 6 experiments, and I get this first mean profile here. This one here. Then I calculate the mean of the second 6 experiments, and I obtain this mean profile here. Note that since the original data was mean-centered, these mean profiles are mean-centered as well. So my factor A, my matrix here, XA, would have the first 6 rows that are identical copies of this first spectrum here, and the second 6 rows that are identical copies of this spectrum here. Then we have the matrix for factor B. Here we have the same situation. So I will calculate for the first spectrum, let's say that is this one, I will calculate the mean of these, and these experiments. So all the experiments and which the factor B is at level 1. From the second spectrum here, I will calculate the mean of these, and these experiments. The experiments where the factor is at level 2. For the third level, I will calculate the mean of these last 4 experiments, and they will be here. Note that also in this case, even when I have 3 levels, 4 levels, these are all centered. If the design is balanced, it means I have the same number of replicates, in this case 2, for each combination of A and B. Then all the matrices XA, XB, XAB, they are mean-centered. This would be particularly useful to understand how we will define that a factor has a significant effect on the response or not. Similarly, I do for the interaction. What is left out after subtracting all these matrices for the original data are the residuals. So this is how the ANOVA decomposition works. Then I have to decide, okay, is this effect significant? How do I deem if this effect is significant. So let's come back here. Let me remove all these drawings that I did. Okay. See, here I have the additive contribution with respect to the mean spectra that is related to the fact that factor A is at level 1. And here I have the additive contribution to the mean spectra related to the fact that factor A is at level 2. Okay? So since they are bound by the design to be mean-centered, well, by the ANOVA conditions to be mean-centered, then, of course, the more my factor induces a significant difference in the signal, the more these two mean spectra will diverge, will be different from 0. And then this is now you understand why to check how to define the significance of a particular effect, we resort to calculate the sum of square of the elements of this effect matrix. Because the sum of square will be larger the larger the difference between the signal ascribable to the different factor level would be. The more this signal diverge from 0, the higher the effect of the factor. So I have now a way, I have made my variance partitioning, and I have now a way to estimate whether the effect of that particular term, be it a factor or an interaction on my response, would be statistically significant. But now, well, first of all, I have a way of defining how to estimate, quantitatively estimate the effect of one controlled factor or interaction on my multivariate response, on my multivariate spectrum. But then I have a second step to undertake. And the second step to undertake is to check, okay, I see that the sum of squares of my matrix element for XA is 100. Okay, but then this variability that I am observing, this difference between these two mean spectra, is it high or low compared to the experiment, to the random error related variability? How do I get a way of testing whether this separation or this separation among the factor level signals is high or low compared to the random error related variability? We do it in a non-parametric way using permutation testing. Permutation testing means that I will check, okay, I got 100 as the sum of square quantifying the effect of my factor A, let's say temperature, on my multivariate spectra. But what would be the sum of square that I obtain under the null hypothesis? So if there is no significant effect of my factor on the multivariate signal. And then the distribution of my null hypothesis, I get it by permuting the labels of my experiments. So here, my true experiments, you see it. First 4 experiments is factor level 1. Second 4 experiment is level -1. Then I keep the same data matrix, but I'm just permuting the label. So I'm disentangling the experimental variability from the design. And I am not doing it a single time, but I am doing it a reasonable amount of time so to have an exhaustive estimation of all the possible, let's say, design unrelated variability. So if I do it a certain number of times, and then I'm calculating the corresponding sum of square of the resulting effect matrix, then I can get an empirically, or let's say non-parametrically estimated distribution of my sum of squares under the null hypothesis. And then I can test the experimentally obtained value with this distribution non-parametrically obtained under the null hypothesis to check whether that particular effect is statistically significant or not. And this is the first part of the answer that I get. So it's the answer that I get, okay, that particular factor or that particular interaction has a significant effect on my response. And then what is the SCA in ASCA? ASCA stands for ANOVA Simultaneous Component Analysis. Well, the SCA of ASCA is that once I have deemed that that particular term is significant, then I want to interpret the multivariate effect of my factor onto the responses. And for doing this, I use simultaneous component analysis that under the ANOVA constraint is simply PCA. So I get this matrix XA, first 4 spectra collected at level +1 of my factor. Second 4 spectra collected at level -1 of the factor. This matrix is made of 4 identical replicates of one spectrum and 4 identical replicates of another spectrum with the let's say constraint of mean centering. So in this case, it would be like that. I take this matrix for interpretation and I do PCA on that. Of course, if I do PCA on this matrix, only 2 different spectra and even more they are mean centered, I obtain in this case a single component explaining 100% of variance. This is level +1 of the scores, and this is level -1. I obtain only 2 score value on this component. Then how do I interpret my effect? Then I have the loadings. Here are the loadings. For the sake of this example, they are just 10 wavelengths potentially, but we will see it very soon on practical example, how it will be with proper, let's say, spectroscopic data. But I have this spectra and these loadings. Okay, what do I see? I see that if my factor A is at level +1, so if temperature is at 30 degrees, I have an increase of these signals with positive loadings and a decrease of these signals with negative loadings. When my temperature is at its lower level, of course, I get exactly the opposite. I will have a higher value of these variables, of these peaks, and a lower intensity of these peaks. So once I get this matrix, like PCA interpretation is straightforward. And that is ASCA, ANOVA partitioning plus interpretation of the effects by PCA. Then, of course, I will skip the mathematics, but then, of course, I can do something that is called the bootstrap, or I could even do jackknifing. And with our real data, this is an example of a spectrum, I can get confidence interval for the loading. So I can even tell, okay, my factor induces a significant variability, not across all the spectroscopic range, but I will just consider these loadings whose confidence interval makes the loading significantly different from 0, so doesn't cross the 0. And then I can even, with this trick, even, let's say, improve my interpretation by checking that only a part of the spectroscopic signal actually has a significant variation induced by the design. A last thing about interpretation, and then I will briefly guide you through a couple of examples that tells us how to interpret this ASCA result. You see here that, of course, if I have 2 levels, I will have just 2 points in the scores plot and a single component to be extracted. If I have 3 levels, I can extract the second components and I will have 3 points in the scores plot and so on. This is the position in the score plot of the mean spectrum for level 1 and the mean spectrum for level 2 or -1 if you want to see it. But I can even check I can even do something that is comparable to what is called multiple testing. So I can check, okay, let's consider here that I have 3 levels, like in this example here. And I want to see, okay, I observed like my sum of squares here, sorry. My sum of squares here tells me that I have a significant effect of my factor. But this significant effect of my factor tells me that the signal at all the 3 or 4 or 5 different levels of my factor are all different. Or for instance, that is a difference between the mean spectrum at level 1, for instance, at 20 degrees of temperature, and the spectra at 30 and 40 degrees, but the spectra at 30 and 40 degrees are not different among one another. How can I get such sort of, let's say, multiple comparison translated in the multivariate framework? I do it by back projecting the residuals onto the model, PCA model calculated for that particular factor. And for instance, here you see in this example that the clouds corresponding to the within level variability are smaller than the difference between the
Sorry, Federico, we cannot hear you. You can please check your connection.
Hello, where did you, I think like you missed the last minute probably, or?
Yes.
Yes, yeah.
Okay, perfect. Sorry, I will share again. So I was telling that as an example, I will show you the effect, a study where we wanted to check the effect of 3 factors, so egg concentration, drying time and temperature on the NIR signal of egg pasta that was produced in the lab according to this design. 5 different replicates were made, well, 5 different pasta pieces were produced at each of the 54 combination of time, temperature, and concentration. Spectra were applied in reflectance, transformed to pseudo-absorbance, and subjected to SNV, and then we did ASCA on that. So here you see the significance. So we found that all three the main factors were having a significant effect on the spectroscopic signal, but also we found the significant effect of the interaction between egg content and temperature. This was particularly important for us because it actually confirmed that due to this interaction, there was a non-linearity in the relation between egg content and, well, the NIR signal and the egg content, and this guided us in a further stage to use a non-linear model instead of standard PLS for making a model for the quantification of egg in egg pasta. But let's see the effect of the different factors on the spectra. In particular, here you see the effect of temperature, according to what I was showing you. Effect of temperature tells us the higher variability is mostly progressive variability along PC1. So most of the variation, almost 100% of the variability, was due to variation along PC1, and variation along PC1 was considered as a progressive variation from negative scores to positive scores corresponding to the temperature. In this case, back projection of the residuals here was suggesting that all the three levels, so all the three different temperatures identify a very different spectroscopic signature. In this case, these peaks with negative loadings have higher intensity when the pasta is dried at a lower temperature, and this one have higher intensity when the pasta is dried at higher temperature, and of course vice versa occurs in moving from lower to higher temperature. So in moving from lower to higher temperature, this wave number decrease in their intensity, and this wave number increase in their intensity. When we consider the effect of drying time, actually here we see a different pattern in the scores. We see a different pattern in the scores. First of all, like, it's true that the higher variability is still along PC1, even if there are some slight small contribution also from PC2. But what we see here by checking for the multiple comparison is that there is a marked difference only between drying at 4 hours with positive load scores and drying at higher time. So it's not worth to go over 6 hours of drying because there is no significant difference after that time. And interpretation is exactly similar with respect to what we saw. I will just skip this example and go to this other example. It was a very complex study involving control of different factors. There was a series, it was a study about honey and characterization of honey using NIR sample done with the group, or mostly done by the group of Professor Marina Manley in South Africa. So the idea was to characterize the effect of storage conditions on authentic honey samples and to check the effect of irradiation and different kind of adulteration on the spectroscopic signals of honey kept at 25 degrees for this other sub-design. And for each of these conditions, also the effect of time was considered. You see the two studies for the storage, only the two main factors were considered to be statistically significant, so temperature and storage time. And for the adulteration study, actually irradiation was considered not to be significant, and only adulteration time and their interaction was considered, their binary interaction was considered to be significant. Here I wanted to show you that when we have, for instance, a time evolution, we can plot the scores as you saw here. So in a traditional scores plot, but we can even take the scores mean value and their variability due to the back projection of the residual and plot it in a longitudinal way. For instance, here, when we consider the storage study and we want to investigate the effect of time on the signal, then you see that we find one component, first component that characterizes a monotonic variation, and another component that characterizes a time evolution where some peaks reach a maximum or a minimum depending to the loadings, and then after a certain time they go back to their, let's say, initial level as a sort of going back to a sort of homeostasis. And here you see the interpretation in terms of loadings, associated load. So I will skip this, and I will just tell you that I'm hoping that I gave you an overview of the potential of the use of ASCA as a multivariate tool for the analysis of design experiments in order to analyze, interpret, and apportion the variability of spectroscopic data collected according to an experimental design. So far, we've just stuck to full factorial design and cross-factor factorial design schemes, whereas the same consideration can be applied to nested design and even in recent time also to mixed model and other complicated, even repeated measure design. So with respect to just taking the data, I'm doing, for instance, a simple PCA, of course, taking the experimental designs into account gives a more straightforward interpretation, but also a more complete and validated interpretation about the effect of the individual terms and interaction on the multivariate data. As it was already mentioned, I will have the pleasure, together with the Italian NIR Society, to host the next NIR 2025 international conference. The website is already up and running, and we hope to see you very numerous in Rome in June. All the details you can find on the website that you see there. That will be regularly updated. Actually, now we have a Facebook and Instagram and a LinkedIn page also that are continuously updated. With this, I thank you for the invitation, for your attention, and I'm open for questions.
Yeah, thank you, Professor Dr. Marini. It was really an excellent presentation, and in a very lucid manner, you have explained how this ASCA can be. It's a very, I mean, using some standard tools like PCA, it can be used for interpretation of NIR data, and that's really straightforward. So I think there will be many questions, but I'm not sure how I mean the participants here are prepared. So any questions from any participant? I have some. So let me take the questions from others. Yeah, Rahul. Rahul, we cannot get your question.
Okay, but there was a question in the chat.
Yeah. Yeah.
So the thing is, well, for that example is, I mean, this is well, the larger variability in that case is quite expected because some of the peaks were the peaks associated to water. So of course we were following. So of course with increasing temperature there was a most effective removal of water. So that were some of the absorption and the wave numbers that you could see. Of course, there can be the possibilities also of some degradation of specific compound, but by the time we were not able to ascribe or to interpret exactly which compound. Of course, due to the nature of NIR, you see like the bands and the transitions that we're interested in. But of course, it was not exactly only using NIR possible to tell which compound possibly could have changed or degraded. For sure, like the OH stretching and the OH combination bands were clear, and that was, of course, something natural to expect with the drying process. Okay, in case of biological sample, the spectra are already even now. Well, the thing is that, yeah, biological samples are. Well, I did actually mostly studies with NIR, with NMR, sorry, biological samples, and in this case, ASCA, even after some days, was giving interpretable results. Didn't have the occasion of analyzing biological samples with NIR, so I cannot answer you on this question properly. The thing is that, in general, the idea would be that you are checking how the variability ascribable to the design is higher or lower than the variability that you have within similar experiments, so within the different levels of the factors. So probably you can still check whether the variability that you may observe due to the design is higher or significant with respect to this variability that you observe within the hourly experiments. And then this is what I can tell about it.
Yeah, I think it was really an excellent presentation, and I just have one or two, comments or some queries regarding the, not exactly on ASCA. It's on the other other chemometric methods that we are now using for analyzing the spectroscopic data, some advanced, some neural network methods, and so on. So, how do you, compare this ASCA methods with this? Just if you can give a general idea on that, or this ANN, or the deep learning methods that are being used nowadays.
Well, I mean, this is a different answer that you get. So I think that, okay, it's quite a rich question to be answered. What I would say is that also there is not a single neural network now, and deep learning. I would say that for the spectroscopic data, probably the one with the highest potential are convolutional neural networks. But the thing is that the variability that we are observing in our spectroscopic data, or even the quantitative relation that we are looking in our spectroscopic data, are some, let's say, really in many cases linear or slightly nonlinear. So there were a study, for instance, by the group of Tom Fern, and that was really nice because he showed that with convolutional neural networks, he could obtain a pre-processing, like he didn't have to optimize the pre-processing. He could blindly obtain a pre-processing of his NIR data that was giving comparable results with respect to him trying to optimize the pre-processing and then doing the calibration with PRS. In that case, for instance, it was funny because he could show that the convolutional neural network blindly based only on the data, was suggesting a pre-processing similar to first derivative. The thing is actually that deep learning requires a lot of samples. So, and a lot means more than thousands of samples. So, and sometimes we really don't need that kind of nonlinearity for our data. We did, with some colleagues, Jose Manuel Amigo, Punit Mishra, Aoife Gowen, and many others. We did a sort of Jean-Michel Roger, a perspective paper recently about the hypes and the pros of, and also cons of using deep learning for NIR spectra. There are things where deep learning can be useful. There are many aspects in which deep learning is oversold. This is my opinion. Also, because really, in order to get the advantage of deep learning, you really need to have many samples covering a higher amount of variability.
No, you're absolutely correct. Our experience is also exactly like that. In most of the cases, the data are linear, and it can be NIR data, and that can be interpreted very easily. And this we can understand. It's also it's much better in case of deep learning or other methods. In many cases, it's very difficult to understand what's going inside it.
Yeah.
Anyway, is there any other question? I saw another one in the chat box.
Okay, there are many questions, so I will go through the questions that are in the chat box. I start from the second. Any idea of polarization effect? Actually, I do not have it concerning with respect to this kind of data. So this is the easiest question answer that I can answer. I will go first. I mean, so one question is that chemometric methods to improve the limit of detection of NIR spectroscopy for low concentration detection of harmful compound present in food. Well, the fact about limit of detection, of course, it's chemometrics can help up to a certain point. So, of course, when we are speaking about NIR, the thing is that, first of all, we have to resort to what we measure. And the instrumental, so in that case, of course, NIR signal is not catching quite well lower intensity, lower concentration compound. What we can aim in these cases is always the same thing, that we are doing an indirect calibration. So mostly, in many cases, we are not quantifying the lower concentration compound itself, but the effect on some other wavelength, some other correlated compound correlated to the one that we are measuring. So if you want to measure something at 10 to the -6 or 1 ppm or so, or to even 10 ppm, you're actually probably calibrating some other things that are indirectly correlated with that. So the thing is that anything that goes in that direction goes in the direction that you have to be sure that that correlation you are observing even in future samples, and that can be quite dangerous. Yeah. For any samples classification based on phorology, it is possible to analyze that data. Yes, of course, it is possible. I would say that probably if you have a reasonable amount of samples, class modeling techniques or discrete or anyway classification techniques would be better than using ASCA. So ASCA, normally you would use it as an say as an unsupervised model or semi-supervised model in the case where you don't have enough sample per class. So if you have like 3 samples for eucalyptus honey, 3 samples for a citrus honey, and 3 samples from a wildflower honey. Of course, you cannot set up and validate a classification model, but you can still check whether the difference in the floral origin produces a significant difference in the spectral signature. That's it. I hope that my answer was satisfactory.
Yeah, it's absolutely correct. No, I think there may be many questions, but at the same time, Dr. Marini is with us for more than 1 hour. It's almost 1 hour 15 minutes here. So I think on behalf of this ANS organizing team, we must, express our sincere gratitude. And there may be many questions. So I request all the other participants, you can send the questions to me, and I can forward it to Dr. Marini. And another point is that since he will be available on during our symposium here in Kolkata, so if you
Yes.
come here, you will have the direct interaction. And I hope that many of the participants here, they are not really very strong in mathematics because they are from different backgrounds. So if you can attend a tutorial of Dr. Marini with some background preparation, I think that will be excellent for your future. So I think with that, let us conclude the session today. Otherwise, it will go on continuing. I request all the other participants, please send whatever queries you have to me. I'll share it with Dr. Marini, and so that you can get the answer within a short time.
So
So
thanks. Sorry.
yeah.
Yes, sir. Actually, I was thinking that you should make some announcement regarding the conference abstract submission and the travel grant for the students. So,
Just before we conclude, Dr. Marini, this our this thing, Shubhadev, you can also announce on our behalf.
yes, actually, in our conference, we have extended the deadline for abstract submission, which is now 31st August. Okay, so I have shared the link for abstract submission in the chat, and also for the students, and we have a waiver program, fee waiver, and your travel grant. So I we will like that you send us abstracts in large numbers, and if it is get accepted, you will get a 50% waiver in your fees. So that's what like I wanted to convey. Thank you.
Okay, thank you, Shubhri, for this announcement. This is for our ANS symposium, which will be held in December. So I think with that, we conclude our session today. Dr. Marini, you are saying something?
Yeah, I wanted to thank you once again for the invitation, and also the attendees for attending and their interest and staying up to the very end.
Yeah, you immediately agreed. It's really nice for us, and we'll definitely welcome you when you are in Kolkata, or maybe on 7th here.
Yes. So see you in Kolkata.
Yeah, sure. Okay. Bye then, and bye all the participants.
Yeah.
Please.
Thanks to all of you and Professor Marini. Thank you, Rajiv sir.